Welcome to Lecture 16, where we will explore the immune system. Adaptive immunity is very important in helping the body fight infection. There are two main pathways that were discussed that can be classified as the humoral response or cell-mediated response. When the response involves the B cells, this is classified as a humoral immune response. In this type of response, antibodies can be produced by the B cells that help neutralize and destroy pathogens or any toxins that are present within the body fluids. The cell-mediated immune response involves the T cells. In this immune response, specialized T cells can help recognize and destroy any infected host or cancerous cells. However, both of these responses can be initiated by helper T cells. Antigen-presenting cells, such as dendritic cells, macrophages, or B cells, can present the antigen to helper T cells using a class II MHC molecule. This will allow for binding to an antigen receptor on the helper T cell. Specifically, there will also be accessory CD4 proteins that will help to stabilize the antigen and receptor complex. The interaction of the class II MHC molecule, the antigen, as well as the accessory CD4 proteins when the antigen binds to the helper T cell will cause the release of cytokines from both cells. This will help to stimulate the proliferation of more helper T cells that are present in the active form. This diagram illustrates the role of helper T cells in immune response. Note that the pathogen is detected by an antigen-presenting cell. This antigen-presenting cell, whether it be a macrophage, a B cell, or a dendritic cell, will have the class II MHC molecule on the surface. This class II MHC molecule will present the antigen to the helper T cell. Note the presence of the CD4 accessory protein that will help maintain this connection and stabilize it. Once this binding occurs, there will be a release of cytokines from the antigen-presenting cell as well as the helper T cell. This will then cause the cloning of activated helper T cells. These helper T cells will then go on to stimulate humoral immunity as well as cell-mediated immunity. Thus, it will activate both B cells and T cells to cause a heightened immune response. The humoral immunity, as stated before, involves the activation of B cells. These B cells will produce memory cells for immune memory, as well as effector cells that will secrete the antibodies to destroy the pathogen or to neutralize the toxin. The activation of B cells also involves class II MHCs on B cells and also involves antigen receptors on the helper T cell. Typically, multiple antigen regions or fragments bind to different epitopes to allow for a range of antibodies to be produced against a common pathogen. As a result, even if the pathogen mutates, it will not be able to evade the immune response. Take a look at the diagram below to see the activation of B cells. Note that once again we have the presence of an MHC, class II MHC, that is present on the B cell. Once an activated helper T cell binds to the B cell, this would cause the B cell to become activated and produce clones that will differentiate into either memory cells that have the antibodies present or effector plasma cells that will then secrete the antibodies that will then be allowed to circulate in the body. Once antibodies produce, they can attach to pathogens or they can interfere with the pathogenic activity. They can do this in primarily two ways. Antibodies can neutralize the pathogen by binding to surface proteins to prevent their attachment to host cells. Note that they can also neutralize toxins in body fluids, or they can also prevent the entry of the pathogen into host cells. Antibodies can also lead to opsonization. This occurs when the antibody binds to surface proteins on the pathogen and helps to form pathogen aggregates. These will then be easily uptaken through phagocytosis by macrophages or neutrophils. Remember, once a pathogen is phagocytized by these immune cells, a more coordinated and targeted immune response can then begin. The cell-mediated immune response involves the activation of T cells. In this case, cytotoxic T cells have the ability to kill any infected host or cancerous cells. In this instance, the host cell will present an antigen on the class I MHC protein. This will then bind to the antigen receptor on the T cell. In this case, a CD8 accessory protein will help stabilize the binding of these cells. Once this binding occurs, the cytotoxic T cell will release two important compounds, perforin, which will help to create pores in the infected cell, and granzymes that will help to break down the infected cell. This will then cause the apoptosis or cell death pathway in the infected cell. This diagram here summarizes the similarities and differences between the humoral immune response that involves B cells and the cell-mediated immune response that involves the cytotoxic T cells. Note that both of them can be initiated by the antigen-presenting cell interacting with the helper T cell. Also note that the antigen-presenting cell can interact directly with a cytotoxic T cell. Antibodies in general will help to neutralize extracellular pathogens or to increase their uptake by increasing the action of phagocytic cells. Cytotoxic T cells, on the other hand, work against intracellular pathogens. These cells will bind to and lyse any infected cells. In understanding the immune system response, it is also important to understand the concept of immunization. In the immunization process, antigens are introduced to the body in order to initiate an adaptive immune response and also to initiate memory cell formation, so that the person has the ability to better ward off the infection. Antigens can be derived from the following mechanisms: through inactivated toxins, killed or weakened pathogens, or through the identification of pathogenic genes. However, the success of a vaccine is highly dependent upon outside factors such as distribution of the vaccine, availability of the vaccine, public information, as well as public receptivity. The importance of these with respect to the success of a vaccine is best illustrated with the resurgence that we now see of measles. Measles was essentially eradicated in the U.S. by the year 2000 after the introduction of the vaccine in the 1960s. However, it is now resurging in various areas within North America and specifically within different areas of New York City as a result of a higher proportion of unvaccinated children as well as adults. It is also important to know the difference between active and passive immunity. Active immunity involves an immune response within the body, such as that which we've seen with B cells as well as T cells. However, passive immunity results when antibodies are introduced into an organism. Some examples of passive immunity include when a mother produces antibodies that can pass through the placental membrane to the developing embryo, or it can be passed in breast milk to her offspring. In addition, a patient may receive antivenom from an immunized sheep or horse if they are bitten by a venomous snake. Immune cells are very efficient at recognizing foreign particles or foreign material. As a result, this can greatly complicate transplants, as organs can be rejected or destroyed within the body of the recipient. MHC proteins that exist on cell surfaces are unique and act like a fingerprint. These proteins are actually what are examined when doctors are looking for a donor match. It is important that the MHC proteins are matched efficiently to decrease the chance that an immune rejection response will occur. In addition, if a person does get a transplant, they are commonly prescribed immunosuppressants. This will decrease their immune response and allow the body to accept the tissue. However, there is also a downside to this, as it will leave them vulnerable to further or other infections. Blood cells, in addition, have specific blood type markers for blood types A, B, and O that consist of carbohydrates that exist on the surface of the blood cells. The body knows to accept surface markers on the innate blood type, and as a result, they will create antibodies against any other blood type. This is because many bacterial epitopes are very similar in structure to those seen on blood cells. Thus, there seems to be a protective mechanism for this type of response, for immediately producing antibodies against your own blood type, or against other blood types that are not your own. If incompatible blood types are indeed mixed, blood cells will be attacked and will lyse, which will cause fever, shock, and eventually kidney failure. Thus, it is important to match the correct blood type in any type of blood transfusion. Immune system responses can also be hyperactive or hypersensitive, as in the case of allergies. Allergies exist when there is a hypersensitivity to specific allergens. This response typically involves IgE or immunoglobulin E class antibodies. In this instance, some IgEs can remain attached to mast cells that are present within connective tissue. When allergens, such as pollen in this instance, binds to the IgEs, the mast cells will release histamine, and these signals will cause inflammation. This can lead to coughing, sneezing, runny eyes, smooth muscle contractions in the respiratory tract. Antihistamines can help in blocking the histamine receptors to help lessen this response. If severe allergic reactions occur, this may lead to anaphylactic shock. In this instance, chemical signals trigger inflammation that cause two important things to occur. Bronchioles will constrict and vasodilation of the blood vessels. This will cause difficulty breathing in addition to an extreme drop in blood pressure. This may result in death. Epinephrine is a drug that can help to counteract this response. It does so by constricting the blood vessels and also by reducing swelling in the throat, to relax the bronchioles and to aid in breathing. Autoimmune diseases occur when a person's immune system basically turns on themselves to target their molecules within their own body. This occurs in the case of lupus, type 1 diabetes, as well as rheumatoid arthritis. In lupus, the immune system can generate antibodies against histones and DNA that are naturally released as the cells degrade within the body. This can cause a systemic response and extreme rashes, as well as nervous system damage. In type 1 diabetes, antibodies can target the insulin-producing cells in the pancreas to cause the inability to produce insulin. In rheumatoid arthritis, the immune system can attack the synovial lining around the joints. This can cause painful inflammation and damage to the cartilage as well as the bone in the infected joints. In addition, the immune system can also be affected by your physical state. Stress and overexertion can lead to more frequent infections as well as more severe symptoms. For instance, extreme exercise can lead to damage to lung and muscle tissue that may make those tissues more vulnerable to different infections. In addition, stress can disrupt hormonal, nervous, as well as immune systems. It is important to note that proper rest has been linked to lower infection rates and shortened healing time. As a result, it is very important that after any major surgery is undertaken, that you take the proper time to rest so that your body can heal and you can get back to normal functioning as soon as possible. It is also known that a healthy diet and a regular amount of healthy exercise can improve immune system function and also reduce your susceptibility to different types of infection. And finally, immunodeficiencies can result when an immune response is either absent or deficient. This can lead to more severe and recurrent infections and can also increase the susceptibility to certain types of cancers. There are two main types of immunodeficiencies that have been identified: innate immunodeficiency that results due to genetic or developmental defects, or acquired immunodeficiency that can be due to exposure to chemicals, drugs, or different biological agents, such as HIV. Both low and overactive immunity can be dangerous for those with COVID-19 infections, making the symptoms more severe. Individuals that are immunocompromised or have breathing issues are highly susceptible to severe infections. There are also adaptations that pathogens may evolve that help them to evade our immune response. Some pathogens can vary their epitopes to limit antigen binding. This is termed antigenic variation. For instance, the parasite that causes sleeping sickness can vary and change the glycoproteins on its surface to easily evade the host's immune response. In addition, the flu virus can undergo frequent mutations and lead to the development of new strains. This is the reason why new vaccines need to be developed each year. Some pathogens can infect cells and then go into a latent period, thus evading the immune response when they are in latency. This occurs in the herpes simplex virus, or HSV. It can remain dormant in neurons, and usually an individual is subjected to an outbreak when their host immune system is compromised with other infections or with stress. HIV is also known as a human immunodeficiency virus. HIV can attack and evade the immune system. Thus, it has a twofold way to increase its persistence within the body. First, it can infect helper T cells by binding to its CD4 accessory protein. It can also infect other cells that display the CD4 protein, such as macrophages as well as brain cells. Once cells are infected, reverse transcriptase will be released from the pathogen to copy out the viral RNA and copy it into a DNA fragment. This DNA fragment will then be inserted into the host cell genome. This will cause the infected cell to produce more viruses. It can also evade the immune system because it has a high mutation rate as well as the ability to vary the cell surface proteins. Prolonged HIV infections can eventually lead to AIDS, which is acquired immunodeficiency syndrome. This is due to ineffective helper T cells. This makes the body more vulnerable to other infections. As a result, people that have AIDS don't usually die as a result of AIDS themselves. However, they typically die due to opportunistic infections such as pneumonia. This chart depicts the immune response in HIV leading to the development of AIDS. Note that the relative anti-HIV antibody concentration after initial infection remains quite high. However, you can see that the helper T cell concentration decreases due to the HIV infection. As a result, the number of HIV particles will increase within the infected individual. If left untreated, this could lead to the development of AIDS. And finally, some pathogens are also known to increase the risk for cancer development. Approximately 15 to 20% of human cancers are actually due to viruses. Some examples are Kaposi's sarcoma herpes virus, which can lead to the development of skin cancer, hepatitis B that has been linked to liver cancer, HPV that has been linked to cervical and oral cancer, and the Epstein-Barr virus, which has been linked to lymphoma and stomach cancer. This HPV viral particle, as seen below, has specific capsid proteins that are identified in yellow. These are the important antigen that are used in current vaccinations. Thus, by knowing the structure of the virus, modes of infection, and how it can persist within an immune system, we can then take advantage of these characteristics to develop effective vaccines.